In this talk, I'll be discussing our work on generating explainable and effective data descriptors using relational learning. And we'll be discussing a case study where we applied it to cancer biology. Although this work is not centered around neural nets, they are worth mentioning. And that's because they've become very popular recently, and that's largely due to the fact that they're very good at learning representations. So we build this very complex network so that we can learn representations that are capable of generalizing to future input data, which just only goes to underscore the importance of having very good representations when building a machine learning model. So the problem we are facing is not unique and is very common in the data science scene, in that we have a set of gene expression data from the library of integrated cellular signatures, and it's for 978 landmark genes. And this data is, like I said, gene expression data. The problem though is that while we have the responses, that is, we have the actual gene expression values, we do not actually have the perturbation conditions that produce these values in input form, that is your traditional CSV file that you may use in your model. However, they do provide the metadata which describes the perturbation conditions that generated the value. So for example, we get the cell line, the cell site, the drug that was used, which is what we most care about, the dosage of the drug, and the time point at which it was measured in hours. So in this case, what we care about is to build a representation of this data that we can use in building machine learning models. So the typical approach of doing this is to use one-hot encoding, right? Which is what we're going to do. And like I said, what we really care about are the drugs, but we'll talk about that next. But what we did was, so for the other input values like cell lines and dosages, we simply, like I said, use one-hot encoding. So based on the cell line, if it's a cell line, we assign 1 if we know what it is and 0 for the rest of them. And the same thing for dosage and every other thing we care about. But since the drug is what causes most of the observed variance we see in the expression values, this is where we paid most attention. So in this case, what is usually done in the literature is that these drugs are represented as Morgan fingerprints, which is what we have here. They're essentially binary vectors that says whether or not a particular chemical structure is present in the drug. And that is the first and typical approach here we considered. So the drug fingerprint approach. But there is another way to go about this, which is the relational learning approach. Because we know that we have additional data for this drug. So for example, we have data from SNAP based on a group in Stanford. And then we also have gene ontology. We have the known pathways they target from Reactome. We have additional data from drug bank, so on and so forth. And all of these things can play a crucial role in building a more robust, or hopefully a representation that we can use to augment our predictive accuracy going forward. So how do we do this? So we take all that data from this previous slide. So we went and we took all this data and then we formalized it in logic, right? As logical rules describing the different relationships between the different entities. And what we're interested in learning here is, what do we know about a drug relating to everything else from the databases I just showed? So we went ahead and we formalized all of this as background knowledge in Prolog. And then using a relational learning algorithm called WOMA, we are able to learn these sort of relations. So instead of having binary input vectors in the traditional sense from Morgan fingerprints, we have these relations. And it is worth noting that these relations are, quite frankly, a lot easier to understand than what you get from your Morgan fingerprint. So for example, this rule here, which is one of the rules that we learned and is actually in the paper, says that a drug has a a frequent pattern in the knowledge you have exists for certain drugs, such that the drug has an oxide functional group and it has two benzene rings. That is something you would not ordinarily get from a Morgan fingerprint, and this is something that someone that is interested in knowing the underlying structure of the drug can easily get at just by looking at the frequent patterns that we learn. So how do we use these patterns in our machine learning problem? Simple. One hot encoding. So just like the Morgan fingerprints, we then go and say, well, we have these relations, right? We then ask for each drug in our knowledge base, which of them are covered by this relation. And if a drug is covered by the relation, we assign a 1. If it's not, we assign a 0. And that's how we form the relational input vectors for the drugs. In the end, we wind up with two input data sets. So one that is based on fingerprints and one that is based on the relations. In our evaluation, we, like I said, use data from links. And then we, instead of looking at all 978 genes, due to computational expense, we preselected 14 cancer-related genes using their dissimilarity. That is, we're interested in seeing how well the method did on a broad range of genes. We used 7,000 samples in our training set and 3,000 test samples, and we considered 3 learning algorithms, so lasso, ridge regression, and random forest. It's also worth noting that for the relational features, we learned 1,024 of them. And the same thing for the Morgan fingerprints. We use 1,024 Morgan fingerprints in our evaluation. So what are the results? What we found is that in general, when the fingerprints are compared to the relations, the fingerprints outperformed the relational dataset on all 3 learners. And these values are statistically significant because we performed pair t-tests and they showed that the fingerprint dataset significantly outperforms the relational dataset or the dataset based on relations. Then we compared a case where instead of using each dataset independently, we concatenated them. So we concatenated the Morgan fingerprints and the relational or relation features, drug features together, and we use it in building models. And we see that things are slightly different in this case. We see that the relational dataset by itself tended to outperform, the fingerprint by itself, or the fingerprint plus the relations, outperformed the relations dataset in all of these 3 cases. But for random forest, it's starting to look like, well, there might be some competition going down the line. In the next case, we compared the concatenated dataset of fingerprints and relations to just the fingerprints, and it's clear here that for even all the 3 learners, the fingerprints outperforms concatenating the fingerprint and the relation dataset on all 3 learners, with the exception compared to the previous 2 results that the difference for lasso is not statistically significant. So we then said, okay, concatenating the fingerprint dataset and the relational dataset or the relations-based dataset doesn't quite work. How else can we apply this? And we decided to stack the predictions made using the fingerprint dataset and the predictions made using the relations dataset. And we found that this stacking procedure outperformed the relation dataset on all 3 learners. And it's all statistically significant as well. So for all 46 genes that we considered, the stacking approach outperformed all the relations, outperformed the relations approach. When compared against the fingerprints approach, we found that just the fingerprints by itself outperforms stacking for lasso and ridge regression. But that is not the case for random forest. For random forest, we see that stacking tended to outperform using just the fingerprints or using just the relational dataset. Lastly, we then compared the stacking approach to the approach where we just simply concatenated the dataset. And we can see that in this case as well, lasso and ridge regression did better when the datasets are concatenated, whereas random forest did better when their predictions are stacked. And the stacking approach we used was just simple averaging. So we took the predictions of the fingerprint dataset and took the predictions of the relations dataset and just averaged them. And we found that here, that for random forest, that tended to do better than concatenating the relations, than concatenating the different datasets or using each dataset individually. So what representation should one use? Well, the results suggest that it depends, because it is clear that the representation one chooses generally depends on the choice of learning algorithm, because as we've seen, the 3 learning algorithms we considered perform differently based on the dataset we used. And secondly, it depends on how the predictions are used, right? So we saw a case where stacking the results outperform just simply using the predictions as they are. So that's why the results suggest that it depends. So one would need to select an approach and how they intend to use the relational representations based on their own specific problem. So in summary, we have shown that we can indeed learn effective data descriptors using relational learning. Secondly, we have shown that these descriptors are understandable, given that they're essentially just simple rules that people can just read and understand directly. They don't need any special or additional analysis to get to the heart of what's going on. The third point is that the choice of learning algorithm and how the predictions are used or how the predictions are integrated impacts the predictive performance, and one will have to choose this on a per learner basis or per dataset basis. And the fourth point is that, and this is really what's stressing, is that even though the descriptors might be understandable, it will not help much if it will not help you understand your model better if your model is a closed black box like a deep neural net or something. It may help if you use a simpler model because you'll be able to analyze it more closely and see what's going on. But if you just chunk all of it into another black box, it will be very difficult to understand. And that is another topic of a different kind of research in terms of model understandability. Thank you for listening.